Guys, I'm so excited to share today's video with you because we're diving into a subject that was my absolute favorite when I was taking AP Biology, cells and their amazing internal structures called organelles. But we're also going to be exploring some of the new incredible developments in this field, including how scientists are engineering new forms of cellular life. A cell is the smallest unit that can live on its own. Bacteria and archaea are prokaryotes with no nucleus, while animals, plants, and fungi are eukaryotes with a defined nucleus and internal compartments. So whether you're one single-celled microbe or a complex human, you are made out of cells. Inside of eukaryotic cells, there are specialized structures called organelles. Some have membranes like tiny bubbles, while others don't. Each organelle carries out a unique job. For example, power production, protein folding, waste disposal, and together they keep the cell alive and healthy. Here are the 6 organelles that we'll be exploring today. Mitochondria, which are powerhouses with their own DNA, nucleus and its internal nucleolus, which is the genome vault and the ribosome factory, Golgi apparatus, which is the cell's post office, peroxisomes, which are detox centers, endoplasmic reticulum, where proteins and lipids are made, and lysosomes, which is the recycling and waste disposal units. Mitochondria have a double membrane and inner folds called cristae that maximize surface area for oxidative phosphorylation, the process that charges up ATP, our cellular batteries. However, without enough ATP, a cell can't do its job. Whether it's a muscle cell helping you move or a brain cell helping you think. Mitochondria aren't just about energy though. They also act as signaling organelles, communicating with the cell's nucleus and other organelles to maintain cell balance and adapt to stress. Mitochondria have their own unique DNA, called mitochondrial DNA, or mtDNA. Unlike nuclear DNA, mtDNA is a compact circular genome inherited almost exclusively from your mom. It includes 13 essential proteins plus RNAs for mitochondrial ribosomes. mtDNA is packaged into nucleoids so it can replicate and express its genes inside each mitochondrion. Mutations in mtDNA or mitochondrial proteins can cause serious mitochondrial diseases, affecting energy-dependent tissues like muscles and brain. Researchers are pursuing gene therapy to deliver correct genes, CRISPR-based editing to snip out mutations, mitochondrial replacement to swap in healthy organelles. The nucleus is your cell's command center. Inside, DNA is wrapped into chromatin and anchored in nuclear lamina. The double-membrane nuclear envelope has hundreds of nuclear pores, gateways for the mRNA transcripts to exit and regulatory proteins to enter, controlling which genes are turned on or off. Moving to the cell's control center, the nucleus, we find the nucleolus. It was once thought to be a ribosome factory, where the cells build its protein-making machines called ribosomes. But now, we know it's a dynamic hub influencing nearly every aspect of cellular life, from how genes are organized to how cells respond to stress and even aging. Even simpler organisms called prokaryotes, which don't have a nucleus, have nucleolus-like structures, hinting at ancient origins for these functions. The way the nucleolus organizes itself often involves a cool process called liquid-liquid phase separation, or LLPS, where molecules group together like oil droplets in water to form distinct compartments without membranes. Next up are the lysosomes, your cell's recycling plant. Lysosomes are membrane-bound vesicles packed with hydraulic enzymes that digest everything from damaged organelles to invading pathogens. Through processes like endocytosis, cargo is delivered to the lysosome, broken down into basic building blocks, and then shipped back out for reuse. In biotech, lysosome function is a hot target for treating storage diseases and even delivering drug payloads precisely where they are needed. The endoplasmic reticulum, or ER, is a vast network of membranes throughout the cell, acting as a major factory for making and folding proteins, as well as synthesizing lipids. It's a critical hub for many biological processes. Sometimes, if there are too many misfolded proteins, the ER can get overwhelmed, leading to something called ER stress. If this stress continues, it can even cause cell death, and contribute to diseases like atherosclerosis, which is a condition that affects your arteries. Golgi apparatus, which acts like the cell's very own post office. It takes newly made proteins and lipids from the endoplasmic reticulum and modifies them, and packages them into little vesicles, and then sorts these packages to send them into correct destinations inside or outside the cell. In neurons, the Golgi is super important for establishing cell polarity, determining which part of the neuron becomes a long axon and which becomes the bushy dendrites. The Golgi can even form outposts or satellites in the distant parts of the neurons, acting as a logical mini post office to deliver crucial materials right to where they're needed for growth and reorganization of neural circuits. About peroxisomes, they're like the cell's specialized recycling and detox centers. They perform vital metabolic functions, breaking down fatty acids and protecting the cell from harmful reactive oxygen species, which can cause damage through a process called oxidative stress. Scientists are discovering how peroxisomes cooperate with other parts of the cell in defense mechanisms, like responding to oxidative stress by releasing an enzyme called catalase. Malfunctioning peroxisomes can lead to severe disorders, highlighting their essential role in health. So, what if we could build life? That's the idea behind synthetic cells and organelles. This involves using concepts from physics, chemistry, and bioengineering to create cellular life from scratch, or to design and construct synthetic cells with reduced but defined complexity. This learning by building approach helps researchers understand how living things work by trying to recreate them. Building synthetic cells isn't just for understanding basic biology. It has a huge potential for real-world impact. Imagine new vaccination strategies, or finding ways to overcome antibiotic resistance. Synthetic cells could lead to new manufacturing pipelines for synthetic materials and alternatives to petrochemicals, which are used to make plastics and fuel. There are two main ways scientists are building synthetic cells. The top-down approach starts with existing living cells and then removes components like individual genes, until it reaches the absolute minimum needed for a cell to live. It's like taking a complex machine apart to see which parts are truly essential. The bottom-up approach, on the other hand, starts with non-living molecules and carefully combines them bit by bit to build artificial structures that exhibit the behavior of living cells. This approach allows for precise control and definition of every component. Synthetic cells is like trying to create life from scratch, and it's really complicated. You need all kinds of skills from biology and chemistry to physics and even computer science. No single lab or country can even pull this off alone. So that's why scientists around the world are teaming up. They're choosing to share tools and knowledge and even educational resources to move faster and smarter together. They're also borrowing ideas from the tech world, like using open access databases and platforms. So students and researchers everywhere can collaborate and learn more easily. It's all about making science more connected, creative, and accessible. So, why is this research so important? Beyond the amazing technological applications, building synthetic cells helps us gain fundamental insights into the very nature of life itself. It pushes us to ask profound questions like, what is life? This field has the potential to shape technology with global impact and improve human well-being. But with the powerful science comes with big responsibilities, like considering ethical implications for biosafety, biosecurity, and how to share intellectual property to benefit everyone. Thanks a lot for joining us on today's video. We hope you had a lot of fun and learned something new. If you like this video, don't forget to like and subscribe for more Biology Bytes. If you care about helping other kids learn, check out Pratham USA. They bring reading and learning to children across India and help build a better world. Visit prathamusa.org to see how you can help. See y'all next time. Bye!